Welcome to the third module of the Working with DNA lessons. This module is about using restriction enzymes and gel electrophoresis. Ideally, you should be working through booklet 3 as you go through the two presentations, A and B. This is the first of the two presentations. This one will introduce you to restriction enzymes. The learning intention of this presentation is to describe the process of cutting DNA using restriction enzymes. By the end of the presentation, you will be able to recall that enzymes are usually proteins that catalyze specific chemical reactions under specific conditions. Recognize how restriction enzymes are used to make recombinant DNA. And identify restriction sites in double-stranded DNA. Please read through this to remind yourself what enzymes are. If you want to learn a bit more about enzymes, check out Sparkhead's enzyme inhibitors lessons. The main uses of restriction enzymes are to make recombinant DNA, which has a range of purposes, and also to find out things like relatedness or genetic disease status. The enzymes themselves are actually very interesting. Originally discovered in bacteria, restriction enzymes appear to have evolved as a defense against viruses. They cut DNA to turn a single molecule of DNA into pieces. We can then separate these pieces using gel electrophoresis. Like other enzymes, restriction enzymes are specific for their substrate. They are so specific that any given restriction enzyme will cut DNA between only particular base pair sequences. For example, this diagram shows where an enzyme called EcoR1 cuts DNA. It works whenever it comes across the sequence GAA TTC. As you can see here, there are many different restriction enzymes which cut at different sequences. You can see that they get their names from the bacteria in which they were originally discovered. Sometimes single-stranded overhangs result from restriction enzyme action. These overhangs are called sticky ends. This makes restriction enzymes very useful for creating recombinant DNA because we can cut a plasmid vector with the same restriction enzyme as the source of our gene of interest, resulting in matching sticky ends. This allows us to then insert the gene of interest into the plasmid vector. Overlapping sticky ends form hydrogen bonds between complementary base pairs. In a moment, it will be a great time to try part A of booklet 3. Before you do that, here's the example sequence in the first question. This is a sequence of double-stranded DNA, and you need to find out where the EcoR1 restriction sites are. So EcoR1 cuts at GAA TTC. So there's our first restriction site, and there's the second restriction site. So this results in 3 fragments or 3 pieces of DNA being produced. The first fragment is 12 base pairs long. The second fragment is 9 base pairs long. And third fragment is a small one at 4 base pairs long. Well done on getting through that. Once again, we hope it was useful. If you have any questions, please don't hesitate to be in touch.